I'm going to be telling the story about the remarkable regenerative capacity of type 2 cells in a human setting. I'm Jaymin from a postdoc from Chapman lab at UCSF. So in a human lung, the alveolar regions, the most frequent cell types that we find is the SPC positive type 2 cells, and the basal cells, 17 or cytokeratin 5, are really restricted within the airway epithelium. However, if you look at some of the progressive diseases like idiopathic pulmonary fibrosis, the lungs of these patients are remarkably different in the sense that the alveolar regions are now replete with basal cells, and there's a reduction of SPC positive type 2 cells. And if you look at some of the more acute injuries, such as H1N1 injured human lungs, and there is even more remarkable expression of cytokeratin 5 positive basal cells out in the parenchyma. And then with the advent of single cell RNA sequencing technique, this particular observation has been extended into larger cohorts of patients. This is reanalyzing one such cohort from coming out of Jonathan Kropsky's lab, which shows that the frequency of alveolar epithelial cells decrease as a fraction of total epithelial cells in an IPF lung as opposed to a normal lung. And conversely, the frequency of basal cells increase in these lungs. And as I said earlier, this is not something that is novel. It's been observed by a number of groups over the past 4 or 5 years. But the question really is, what is the source of these expanded basal cells? And that's the question that I'm trying to address here. There are two really obvious candidates for this. One of them is airway basal cells. Knowing their remarkable progenitor activity, they could potentially get activated, migrate out in the alveoli, and differentiate. However, in our hands, or we are not aware of any evidence that shows this, that a human basal cell can give rise to a human type 2 cell in vitro or otherwise. The second option is the type 2 alveolar epithelial cells. We all appreciate the remarkable regenerative activity of these type 2 AECs, especially in the human context. So the question we wanted to ask is whether these human AEC2s can give rise to anything other than type 2 or type 1 cells in vitro. That we live sorted type 2 cells. This is single cell analysis of these cells, which shows virtually all cells express SPC, no K5 expression. And when we co-culture them with MRC5 cells, which is an embryonic fibroblast cell line, and this is something that has been done before, and consistent with what others have noticed, we also see expression of SPC positive colonies with no detectable expression of cytokeratin 5 positive colonies in this particular co-culture system. However, if we switch the mesenchyme and if we now culture these type 2 cells with adult human lung mesenchyme, we see something remarkably different. Just within the first 7 days of culture, we see that these SPC positive type 2 cells now start also expressing cytokeratin 5 positive protein. And by day 14 in culture, virtually all of these SPC positive type 2 cells lose their surfactant protein C expression and acquire expression of cytokeratin 5, which is a marker for basal cells. So we see this remarkable transdifferentiation of a bona fide adult type 2 cell into a K5 positive basal cell just within 14 days. So to really understand what kind of dynamics are going on, we took help of single cell RNA-seq and of this particular, in this culture system. We start with virtually all type 2 cells. And within the first 7 days of co-culture with adult human lung fibroblasts, we see at least 3 different populations. This is some sort of intermediate cell types that co-express both alveolar and basal cell markers. And we see basal cells. By day 14, we see emergence of SCGB1A1 positive club cells. And by day 21 in vitro, we see emergence of multicellular cells, which express FOXJ1, acetyltubulin, This was really remarkable, and we are trying to understand what are the gene pattern changes that kind of correlate with this particular transdifferentiation. We see that, as we expect, as the cell culture progresses in time, the genes associated with mature type 2 cells decrease, such as SPC, ABCA3, LAMP3, and basal cell markers such as cytokinin 5, 14, and 17 increase. This intermediate time when we see co-expression of number of type 2 markers and basal markers, as well as cytokinin 8, which marks this intermediate cell population. And we don't think this is a discrete, unique population. I think we think it's a transient population that shows up as part of differentiation. So to understand what can drive this particular type of differentiation, we looked at our single-cell RNA-seq, and we found that the BMP signaling is downregulated as a function of time in cell culture, where the airway program by virtue of SOX2 expression and basal cell differentiation by virtue of P63 activity is upregulates. So we wanted to ask a simple question: Can treating these co-culture with BMP4 rescue some sort of type 2 fate? And indeed, we see that when we treat these co-culture with BMP4, we increase the expression of SPC message and downregulate the expression of cytokinin 5 message. There are a number of more mechanistic studies that are done, but with the paucity of time, I can't go in details. So the second question we wanted to ask, is this just a phenomenon that is observed in a one cell type, in an adult type 2 cells? Or can we utilize other models of type 2 cells that can show similar capacity to transdifferentiate? So for that, we took help of Cotton Lab, who are happy to share with us their SPC iPSC-derived type 2 cell line. And in this particular cell line, one of the allele reports SPC expression by the virtue of 3 tomato, and the other allele produces functional type 2 cell, functional SPC protein. So when we put these cells with MRC5, as expected, they give rise to virtually all 3 tomato positive and therefore SPC positive type 2 colonies. However, again, if we switch the mesenchyme and put it with adult human lung primary mesenchyme, we see two things. One, the colonies are smaller, not less frequent, but just smaller. And two, they all lose their SPC expression as judged by 3 tomato expression. And if we treat these cultures with BMP4 and TGF-beta inhibitor, we see, not surprisingly, increase in SPC positive colonies with MRC5 co-culture, but we also see partial rescue of some of the SPC positive colonies in a primary co-culture. So we wanted to focus on this particular system, and we wanted to see what are these colonies which are SPC negative and they are smaller. So we wanted to see if any of them are actually these intermediate cells, if they are stuck in that particular stage. So we stained them with cytokinin 17 and 8, which are two of the markers of these intermediates. And indeed, the vehicle-treated IAC2s co-culture with human lung mesenchyme express cytokinin 17 and 8 without any detectable expression of 3 tomato or SPC protein. And consistent with what we saw with BMP4 and TGF-beta inhibitor treatment, we can reverse this particular phenomenon and show the expression of SPC by 3 tomato expression and baseline expression of cytokinin 8. And if we tweak the culture conditions just a little bit more, turns out that we can actually drive the expression, transdifferentiation of these IAC2s into cytokinin 5 positive and KX2.1 positive lung basal cells without any detectable expression of either 3 tomato or SPC protein. We don't know what are the exact signaling pathways which is causing this. We are actively trying to work it out right now. But then our next question was that, great, we can do this in vitro. Can we do this in vivo? To answer this question, we took help of our transplantation model, in which we injured immunocompromised mice with bleomycin, and 10 days into the injury, transplanted 500,000 type 2 cells into the injured lungs. And about a week or 10 days later, we harvested the lungs. And not surprisingly, our data matched with our in vitro data that showed that human nuclear antigen positive, which is marked by HNA in red, we can see both cytokeratin-5 positive basal cells and SBC positive type 2 cells coming out of the transplanted cells in an injured lung. And not only that, when we further look at these K5 positive clusters of engrafted cells, we also find evidence of club cell and differentiation as judged by the staining of SCGB1A1, which marks club cells, and acetyltubulin, which marks multicellular cells. So we have so far showed that we can drive the transdifferentiation of mature type 2 cells into basal cells, both in vitro and in vivo. And we went back to our original question, which is, can any of this explain, at least partially, what we see in IPF's epithelium? So we looked at the single cell RNA sequencing of IPF epithelial cells. And as I had mentioned earlier, we see very few type 2 cells. Majority of the cells are cytokeratin-5 and 17 positive basal cells. And we also see these smaller cluster that has some remnants of basal alveolar epithelial type 2 markers, and also low expression of basal markers. So with the help of RNA velocity, which predicts a future state of given cell based on the splice to unsplice ratio of its RNA content, we saw that this particular analysis predicted a type 2 to ABI to basal cell differentiation, at least in silico. And this is not just something that we observe in silico, but when we go back to our IPF lung, we see evidence of particular intermediate cell types right at the interface of where you expect to find normal looking alveolar region to some sort of a thickened parenchymal regions with some sort of alveolar structure, and then further much more heavily fibrotic lesions. We see that we can find these cytokeratin 17 positive, ABCA3 positive, SBC low, or in some cases SBC negative, and cytokeratin 5 negative intermediate cells. So this is something, not something we can really manipulate in vivo in human lungs, but combining all of our in vitro, in vivo studies in mice and looking at the IPF data sets, what we are really suggesting is that based on microenvironment, based on some sort of aberrant niche factors, the dysfunctional type 2 cell, or the regenerative capacity of type 2 cells become dysfunctional. And they differentiate into these intermediate cells that can ultimately give rise to basal cells, and really represents a compromised regenerative pathway in the alveoli. And with that, I would like to thank Chapman Lab, especially my mentor, Hal Chapman, and Alexis, who has worked a lot with me on this particular project. Specifically, also would like to call out Pang Lab, Chao Soon, who's a postdoc in Tian Pang's lab. He has given a lot of great mechanistic insights into this process. And Cotton Lab for always providing with their expertise and cells whenever we ask them. Thank you.